What is the BCR-ABL1 fusion protein in CML? Imagine a tiny switch inside your blood cells that is supposed to turn on and off normally. In chronic myeloid leukemia, or CML, this switch gets stuck in the on position, causing cells to grow out of control. The reason for this is a special abnormal protein called the BCR-ABL1 fusion protein. This protein is made when a specific genetic change happens in the blood cells. Normally, our genes are organized on chromosomes, which are like packages of DNA. In CML, a piece of chromosome 9 swaps places with a piece of chromosome 22. This swap is called a translocation and is labeled as T922. This creates a shortened chromosome 22, known as the Philadelphia chromosome, which is a hallmark of CML. This translocation fuses part of the BCR gene from chromosome 22 with part of the ABL1 gene from chromosome 9. The result is a new gene called BCR-ABL1. This gene encodes the fusion protein, which is an abnormal enzyme called a tyrosine kinase. The fusion protein is about 210 kilodaltons in size, often called P210. What makes it dangerous is that it is always active, constantly sending signals inside the cell. This unregulated activity causes the blood cells to grow and divide too much, leading to the buildup of abnormal myeloid cells in the blood and bone marrow. The BCR part of the fusion protein helps it form complexes and interact with other proteins, while the ABL1 part provides the tyrosine kinase activity. This activity triggers multiple signaling pathways that tell cells to grow, survive, and divide. These pathways include RAS, RAF, MYC, and JAK-STAT, all of which are involved in cell growth and survival. Because of this, the BCR-ABL1 fusion protein is the main driver of CML, making it a key target for diagnosis and treatment. Detecting the BCR-ABL1 gene or protein in blood or bone marrow is essential for confirming a diagnosis of CML. It also helps doctors monitor how well treatment is working. Many therapies, called tyrosine kinase inhibitors, specifically block the activity of the BCR-ABL1 fusion protein. One well-known example is Imatinib, which has revolutionized how CML is managed. By blocking this abnormal protein, these drugs can control the disease, reduce symptoms, and improve quality of life. In summary, the BCR-ABL1 fusion protein is a crucial molecular abnormality in CML. It results from a genetic swap between chromosomes 9 and 22, creating a fusion gene that encodes an always active enzyme. This enzyme drives the uncontrolled growth of blood cells seen in CML. Understanding this fusion protein has allowed scientists and doctors to develop targeted therapies that have transformed the outlook for many patients with this disease. Always remember, if you or someone you know is dealing with leukemia, consulting healthcare professionals is vital for personalized care and treatment options.